Congratulations, you've gotten a raise to tertiary and quaternary protein structure. We've been talking a lot about primary structure, so our sequence of amino acids, and how that influences secondary structure, which is the backbone hydrogen bonding that gives us things like alpha helices and beta strands and beta pleated sheets. Now we're going to talk about how those can actually join up to form things like motifs, which are kind of like the furniture within your rooms, and domains, which are kind of like the rooms themselves. And the way that they're interacting with one another is going to be through interactions of the side chains largely. And when we talk about the side chains getting involved, well, now we're talking about tertiary structure. And so tertiary structure is going to involve those intermolecular forces and those interactions between the side chains and between the side chains and the backbone of regions of a single chain. And then sometimes proteins are made up of multiple chains, in which case we can talk about quaternary structure, which is going to be interactions that happen between the different structures, as well as also like just kind of like the whole overall structure itself if you have multiple chains. That's when we're talking about things like oligomers and multimers and all that good stuff. So let's dive in. So we have those 20 main amino acids that hopefully you all know really, really well by now and know all of their unique properties. So the different amino acids all have these different properties. And therefore, when they link together to form proteins, the proteins are going to have the properties of those amino acids. And the properties of the amino acids are going to influence how that protein folds up and thus functions. And so we saw how we can combine amino acids through peptide bonds, and how these peptide bonds were going to be resonance stabilized, and thus they were constrained to planes, where we could only get rotation at those certain places, those phi and psi angles in between the planes. And remember, we call those angles dihedral angles because that's the name for an angle between planes. We also saw how the amino acid itself, the identity of the amino acid, could also further limit the rotation, because if you have something big and bulky, say, it's going to clash if you try to rotate it certain ways. Also, because of the backbone having that same generic part, in addition to all of those unique R groups or side chains, you have that generic peptide backbone, which has hydrogen bond donors in the form of the amine groups, and hydrogen bond acceptors in the form of those carbonyl oxygens. And because they're evenly spaced out along that backbone, and because those dihedral angles are constricted to certain places, you get these kind of characteristic secondary structures. In all sorts of different proteins, you can get these same things, these beta strands, and these which can team up to form beta pleated sheets, and these alpha helices. And so we looked at that a lot when we talked about secondary structure. And now it's time to move on and see how these secondary structure elements can actually combine, such as what you're seeing in the middle, how they can combine to make tertiary structure. And with tertiary structure, here we're saying, okay, we're not just going to think about the backbones. Now we're going to directly think about the side chains. So we had kind of been indirectly thinking about the side chains because we were thinking about how they would influence the secondary structure. But when we were talking about the secondary structure, the interactions we were actually talking about, like the IMFs, those intermolecular forces, those are going to all just be like backbone interactions. That was going to be the hydrogen bonding between the backbone. So when we talk about the secondary structure, we're talking about backbone, H bonding, and typically within parts of the protein that are like close by in sequence. Although in the case of the beta strands, you can also get things kind of loop back and connect, even though they're further away in sequence. We also saw how those are the kind of like two main secondary structures, and we also had like beta turns. So we have that'd be beta turn. And basically those are kind of like the regions that are going to connect the strands. So those are the main types of secondary structures, but then we also have regions of proteins without secondary structure, and these are called our intrinsically disordered regions. No secondary structure. So basically these regions, there's not forming hydrogen bonding between that backbone. You don't have things set in like, or at least not significantly, you don't have things set in place. Sometimes these regions can actually come and take shape when they bind something, but often they are more just still flexible regions that can serve as scaffolds and things. Some proteins have a lot of IDR, like almost all RDR, and these might be good for things like phase separation. And then other proteins might have just little regions of intrinsic disorder in between, say, those more set in stone secondary structures. But that would be your IDR. So now let's move on. We're going to talk about tertiary structure and quaternary structure. So remember, tertiary structure, this is going to involve our side chain interactions. We can have side chain backbone interactions. So we have side chain. And remember another word for that side chain, we're talking about the R group. Those are like terms for the same thing. We can talk about side chain backbone. We can talk about things like side chain cofactor, which we'll talk about. So something like a small molecule that it's bound to, various things when we're talking about the side chains getting involved. And then quaternary structure, not all proteins have. So we have only some proteins. And basically, this is going to be if you have multiple chains. So remember that when we make a protein, when we make them as polypeptides, so these long chains. And then some proteins, they have multiple chains. So you would get things like multimers where you have maybe a dimer or a trimer. We'll talk more about these later, but these can be the same subunit or they can be different subunits. So let's look at what kind of interactions we could get in our tertiary structure. And these are the same sorts of interactions that we could also get with our quaternary structure. Just in the terms of the quaternary structure, the interactions are between different chains, whereas in the case of the tertiary structure, the side chain interactions are between, and the side chain backbone interactions, those are all between the same places in the same chain. And so the interactions that can form, well, those are going to depend on the amino acid makeup. And so we talked about how we have those 20 main ones, and we talked about how they all have different properties. And so we have ones that are nonpolar, some that are polar, some that are plus charged, some that are minus charged, and various things like this. And we discussed the types of interactions that they can form, and we'll go over these again. But we also have to think about not just these 20 ones, we also have to think about how they can be modified. So basically, you can get what are called post-translational modifications. So remember that translation is the process of making a protein by piecing together those amino acids through those peptide bonds. And this is done by using the genetic information that's held in the messenger RNA or mRNA. So that's the process of translation. And the amino acids are put in place in the form of these, in their original form. But then what's going to happen is that there are enzymes that can actually modify them. They can do things like phosphorylate them. So add a phosphate group, methylate them, acetylate them. There are various post-translational modifications that are going to add extra types of functional groups, extra types of ways for these amino acids to interact. And note that I've been saying like 20 main ones. Basically, these are the ones that you need to know about. These are the ones that our body is going to use. There's one called selenocysteine, which looks like cysteine, but it has a selenium instead of a sulfur. And so we use that very, very sparingly. And then some little bugs have, the bugs like bacteria, archaea and stuff, they have a thing called pyrolysine, which is kind of like a weird looking lysine thing. But basically our bodies don't use that. Our bodies use these 20 main ones and they add them like this, and then they can be post-translationally modified. And the significance of that is that they're going to be able to, say, change the function of a protein, alter its activity, add binding sites. Because now if you have if you're changing the properties by, like, say, introducing some big bulky charge thing, well, then we talked about how opposite charges attract and like charges repel. So that could influence the IMFs around it, influence the whole protein shape. Or you could be adding a binding site for something because now you have an ion there, which can then form an interaction. So hopefully you're getting a sense that basically we have, by changing the different functional groups that are available, by changing the properties of these amino acids, we can therefore change what types of interactions can form. And then finally, We can also add extra ways to make interactions by binding to other things that aren't proteins. So we can have this is commonly seen with like enzymes, where we have an enzyme that's like by itself. We call this an apoenzyme. So apo is kind of like empty. And then when it binds to a cofactor, we get like a holoenzyme. And what do I mean by these cofactors? So sometimes they could be metals, sometimes they can be vitamins, or they can be things like other small organic molecules. And so basically proteins don't have to act alone. They can act they can bind to metals, they can bind to vitamins and other small molecules, things that are going to allow them to have additional functionality, additional functional groups that weren't present in those 20 main amino acids. So let's talk a little bit about the types of interactions that are involved in tertiary and quaternary structure formation. Now, these are almost all going to be intermolecular forces, or IMFs, those like non-covalent interactions. So these are going to be mostly non-covalent. There's a single exception that we're going to talk about, and that's going to be our disulfide crosslinks. And we'll talk about them first. Get them out of the way. And these are going to only form between cysteines. And the rest of them are going to be non-covalent. I should also mention that when we say like IMFs, so IMFs, those would be our intermolecular forces. And so typically when we're talking about intermolecular, what we're talking about is like between molecules, right? But in the case of proteins, we kind of use this, we're actually going to be talking about between regions of the same molecule. So just different places on the same protein. So they're not technically intermolecular, but we're still going to refer to them as intermolecular interactions, and the same principles hold. And what were those principles that we have? We had that if you had, think back on Coulomb's law. We said if we had greater charge, that would be greater attraction or repulsion, but we're just going to be talking in the case of attraction, we're going to be thinking about like opposite charges. What else did we say? We said that if we had a shorter distance, that was going to be a greater attraction. And we said that if we had a lower epsilon, so that was our dielectric. So basically that would be lower polarity of the solvent. That's going to make it easier for those, that would make it easier for those forces to feel one another, and thus you get the greater attraction and repulsion. And again, we're just going to mainly be thinking about attraction, but the same thing would hold in case of repulsion. So when we're thinking about what would make a stronger intermolecular force, we want a greater charge, shorter distance, and less polar solvent. So maybe being in the inside, the hydrophobic part of a protein, rather than on the outside, the hydrophilic part, the surface, where it's exposed to the water, assuming we're talking about like a cytosolic water-soluble protein. And so when we talked about the different types of IMFs that we could have, basically we said that our strongest one was going to be our salt bridge. Remember, that's just another term for an ionic, an ionic interaction. So that's where we have full plus minus charges. And then we had our permanent dipole-dipole. Where we had that special case was going to be our hydrogen bonds, which were going to be the strongest one of those, and we'll review those again in a minute. And then we had our just like induced and transient dipoles. So we were talking about like our van der Waals interaction. We had our dispersion forces, our like non-polar interactions. And sometimes they're referred to as like hydrophobic interactions. But remember that the real driving force of the hydrophobic effect, it's not that these hydrophobic things are attracted to one another. They don't have attraction to one another. Intrinsically, they're just forced together because the water excludes them. And once they're forced together, well, now they have the chance to have those transient dipoles come into play and then make them attract to one another. Just temporarily, fleetingly, weakly, but because you have so many of them clumped together, then you can actually see an effect. And so these are going to be the things that we're going to look for. So the strongest of them is going to be the disulfide crosslinks, because these are actually going to be covalent bonds. And so in order of strength, we would have, this would be the strongest. Then we would have like our charge-charge interactions. So we'd have our salt bridges. We'd have our permanent dipoles, and we'd have our induced or transient dipoles. Okay, so now let's go and let's look in these in a little more detail. Starting with those crosslinks. So remember, these are going to be our strongest types of interactions, and they're only going to happen between cysteines. So remember, this is cysteine. It has that thiol group, that sulfhydryl group. So thiol is AKA sulfhydryl. So cysteine can deprotonate to form thiolates, and it can also oxidize to form crosslinks. So basically, two cysteines can join together to form this cysteine. I don't know how you actually pronounce things, but basically, when you think about them losing their H's and coming together, you lose that E in the name. So you go from cysteine to cysteine or whatever. And again, I never really say these out loud, but that's technically what they're called. And we often call these crosslinks. Another name for them is disulfide bridges, and disulfide bonds. Those are all names for this same thing. Now, sometimes this happens on purpose and sometimes it happens on accident. And so it can happen if you have oxidative species, reactive oxygen around. It can also happen in a controlled manner to introduce site-specific crosslinks, because remember, these are going to be our strongest interactions. So these are going to be strong. And when we're talking about strength, remember, we're thinking about like a single one. So a single one of these is going to be stronger than a single one of our other types of intermolecular forces because it's covalent. But remember that when we're talking about strength, we're talking about like of a single one and not thinking about, okay, well, in the case of van der Waals, yeah, they're really weak individually, but when you have a bunch of them together, well, then you can actually get a strong effect, a very, very strong effect. That remember that pages of a phone book being interlaced and being able to pull a truck. But in this case, we're talking about an individual cysteine-cysteine bond, this crosslink. And so this is going to be strong, and therefore it's helpful to kind of help certify proteins, especially that need to be secreted. So basically, often in secreted proteins. And why is this? Well, inside, not only do they need to be sturdier in order to survive inside, like the whole journey wherever they're going after they're secreted, but also this inside the cell, it's kept in a very reducing environment. So basically, we have this molecule called glutathione, and this is going to be our biological reducing agent. So we'll see that in the lab, we'll use things like DTT and beta-mercaptoethanol in order to reduce things. But in our bodies, we use this molecule called glutathione. And basically, it can get oxidized and reduced. And it can reduce the cysteine crosslinks back to their individual cysteines. And in the process, it gets oxidized. And then we have enzymes that can actually go and kind of reset things. So just a quick note is that, like, you might think like, okay, well, why is this actually called oxidation? Well, if you think about what you're doing, when you're just deprotonating it, that's not oxidation because you're not losing any, you're not gaining or losing electrons. But in the case of when you're actually joining up to another cysteine molecule, well, now what you're doing is you're replacing basically a bond to a hydrogen or no bond to a hydrogen, but basically you're not
you have those electrons, you own those electrons. Remember that oxidation is loss of electrons. And so how do you lose electrons? Well, you lose electrons if then you go and you form a bond to another sulfur. And that sulfur is going to then pull some of those electrons away from you. And so that's going to be oxidizing it. Oxidation is loss of electrons or electron density. So you're losing some of the shared electrons because now you're sharing them with the sulfur versus having them shared them with the hydrogen. So review the video on redox if you're confused about that. But basically, we can say that cysteine gets oxidized and forms these crosslinks. This oxidation can happen on purpose or it can happen on accident. And so when it happens kind of like on accident, that can be a bad thing because it can be, if you have like reactive oxygen species around, it can cause this oxidation that you don't want. It can also cause some helpful oxidation, but a lot of times it's things that you don't want. And that's why our cells have that glutathione to maintain the reducing environment. So inside our cells, this is going to be so high inside cells. And this is going to make it so that our cells can protect ourselves from having those unwanted crosslinks. But if we do want crosslinks, like if we want to secrete something, well, then we need a way to put in those crosslinks. And so basically secreted proteins have to have special, they take special pathways. They go through different compartments inside the cells to make sure that they can actually get those crosslinks to formed the correct way, and then have them be secreted while they're still there. Because outside of the cells, it's going to be an oxidizing environment. So inside the cells, you have high glutathione. So you have reducing environment. And then outside the cells, you're going to have an oxidizing environment. So those crosslinks can stay in place. Now, you need to make sure that the crosslinks form the right way. And you probably have a sense about this if you know what's behind a hair perm. So basically, in hair, there's this protein keratin, and we have a lot of this keratin in our hair. And basically, it has a lot of cysteines, and those cysteines can form disulfide bonds with other strands of keratin. And if you have, depends on how these bonds are formed, your hair might be really curly or it might be straight. And because those are like actual covalent bonds, well, they're not just going to like break apart if you say get your hair wet. So you might be breaking up a little bit of the hydrogen bonding, but you're not going to be breaking up those crosslinks. And because of this, your hair might get like flatter or straighter temporarily, but then it's going to kink back into that curl. And the reason why is because you have those disulfide bonds. And those disulfide bonds, well, they're going to be like permanent. But if you break those up with a reducing agent, well, now you can and then you put your hair around a curler and you allow them to reform. Well, now what's going to happen is that you're going to have them form in different places. And this is going to make it so that your hair it will take the shape that it was. So if it was straight or if it was curly, basically these ways you can now have formed new cysteine bonds that give the proteins a different shape and give your hair a different shape. Okay, so now let's think about not just in our hair, but all throughout our body in all sorts of different proteins. What's going to happen is how those cysteine crosslinks are put together is going to really influence things. So when we talk about these cysteine crosslinks, basically we have to think about them as being really specific, or hopefully really specific, because we want these to be formed in the correct way, which is why even when we have functionally important like crosslinks formation, we need to make sure that they're happening in a controlled environment where we make it so that those proteins form the correct crosslinks and not wrong ones. So we actually, in addition to keeping a reducing environment, there's few cysteines inside of proteins. So it's so that there aren't these many places for things to go wrong. So for say there to be that unwanted oxidation, which can then make it more likely to form these crosslinks to things you don't want. And so cysteine crosslinks can play important roles, not only within the same protein, but also between different proteins. So you might see them kind of help hold dimers together, but we'll see also that like a lot of dimers aren't actually going to be held together through crosslinks, but some of them actually are. And you'll see examples of the ones that are for, say, things like antibodies, where the different chains are going to be held together by these cysteine crosslinks. Those antibodies need to be really sturdy as they move around the body, and they're all of that stuff. And so they're held together by these crosslinks. So these crosslinks are going to be strong and specific, but we have to make them in a controlled manner. And if they're typically not found inside of cells because of the reducing environment. And that reducing environment is there to protect our cells. So now let's talk about just like the more generic forces, the weaker forces, the IMFs, are non-covalent interactions. So remember that when we talked about these IMFs, we could kind of simplify things down by thinking of just opposite charges attracting. In the case of a salt bridge, in the case of an ion-ion interaction, well, here you have full charges. In the case of a permanent dipole, such as hydrogen bonding between water molecules, well, that's going to be like permanent partial charge attraction. Then you could also get like temporary dipoles form through induction by having something charged come by and kind of push the electrons away as they get repelled by nearby charge or attracted to nearby charge. And then we can have those instantaneous dipole-induced dipole, basically where things just transiently, because those electrons are all whizzing around randomly, they happen to be in one place more than the other, you get this instantaneous dipole that then induces a dipole in the thing next to it. And if you have a lot of these things clustered together, such as because the hydrophobic effect due to the entropy gained by freeing waters from solvating things pushes all those hydrophobic things together, and then all those hydrophobic things together have these transient dipoles formed. You get these kind of chain reactions that are individually very, very weak, but collectively strong. And so those are the types of IMFs that we're going to be thinking about now. But in terms of which one's the strongest, those would be our ionic ones, right? Because those have full charges. And when we talk about these salt bridges, these ionic bonds, so remember a salt bridge because it's a plus and a minus, and a salt is a cation and an anion that neutralize one another. And so that's what we have here. We have one of our basic amino acids. And then we have one of our acidic amino acids. And remember, when we talk about basic or acidic, that's because we're referring to what it is in its neutral state. So in the case of our acidic amino acids, we have aspartic acid, which in its neutral state is aspartic acid, but we typically find it as after it has already acted as an acid, gave up the protein, and we see it in this aspartate state, its conjugate base. In the case of glutamic acid, we typically see it as glutamate in its conjugate base form, but we name it as acidic because we're talking about what it is. Does it act as an acid or a base in its neutral form? In this case, it acts as an acid in its neutral form. What about our basic ones? Well, here we have lysine, arginine, and histidine. In their neutral form, they act as bases. They take a proton and then they become positively charged. So even though we find lysine and arginine and a fair amount of histidine in their protonated form, most of the time, basically what's going to happen is that's going to actually be the conjugate acid form. And so we're almost always going to find lysine and arginine in their conjugate acid form, histidine sometimes, and we classify them all as basic. And when we have one of them, and those two of them like interact with one of the other type, that's when we get a salt bridge or an ionic bond. Now, because there's only those few options for these charged ones, unless we're talking about something that's been phosphorylated, well, here there's going to be greater specificity. So not as specific. So this wouldn't be not as specific as the cysteine, as disulfide, because there's more options and because they're more common than the sulfur, but more specific than other IMFs. Because there's going to be those fewer options, and they're going to be individually stronger. And remember, they're going to be even stronger in like the hydrophobic center of a protein where the dielectric constant is lower. There's less kind of stuff distracting them. They can feel one another better. And so that would be these forces. And remember also that those forces are going to be able to act over a slightly larger distance because their force of attraction falls off more slowly when you go farther away. And it also because you start with a higher charge. So that all goes back to our Coulomb's law. What other types of interactions can we talk about? So we've talked about those disulfide bridges. And remember, those just formed with cysteine. So we had cysteine. So we could have disulfide, our crosslinks. Remember that those were often going to be in secreted proteins because our cells keep it reducing environments. We then talked about how we had the basic and acidic. So remember, these would be our acidic, and these would be our basic, and these would form our salt bridges, our ionic bonds. Okay, well, what would be next? Well, now we want a permanent dipole, right? So we want something that's polar or uncharged. And so these we can form our dipole-dipole. And often this is including H bonding. And remember that our hydrogen bonding was going to be the strongest of our dipole-dipole because they're shorter, because you have that partial covalent bond character. And remember, for a hydrogen bond, we need to have a donor, which is a hydrogen bound to something electronegative, such as an oxygen or a nitrogen. And then for an acceptor, we need to have some a lone pair on something electronegative, like an oxygen or a nitrogen. And so if we have a hydroxyl group, if we have a carbonyl group, if we have an amine group, all of these have opportunities to act as hydrogen bond donors or acceptors. And therefore, we can form hydrogen bonds with these amino acids and between these amino acids. We can also form hydrogen bonds between the amino acids and the peptide backbone, in addition to those secondary structure backbone-backbone interactions we saw. Okay, finally, what about the rest of them? Our nonpolar uncharged, these can basically form our basic, these can form like dispersion forces. So those just like transient dipoles. And other nonpolar interactions. So other types of VD wall. So basically, these can only do those like transient induced dipole or instantaneous dipole, all those things that are individually weak. Collectively strong. And these are going to be our least specific. So we talked about, we only had a few cysteines, and they had to form under these like certain environments with the help of enzymes, often making things happen the right way. And so we're only going to get those very specific crosslinks. But then we said, okay, well, the salt bridges, those were still going to be more specific because we only had a few options that had full charges. And so those are going to be more specific, but not as specific because we still have those few options, etc. When we talk about our polar ones, well, there's a lot of those. And so not only can those polar uncharged ones form those polar interactions, but so could the other ones that were actually charged. Those can also form these. And you can also get them interacting with the backbone. So basically, those are going to be less specific. But the least specific is going to be the rest, like this nonpolar uncharged, the dispersion forces, other van der Waals forces, because all of them can do that. And so basically what can happen is that these are going to be not only the weakest, but the least specific. But remember that the weakest refers to in isolation. And so this is why we can get those big effects when we push them all together. And the big effects come not because they're weak, not because they're strong, not because they're attracted to one another, but because of the circumstances. They're all kind of like just pushed together. Remember that the driving force of the hydrophobic effect that actually pushes them together isn't a desire to bind to one another. It's the gain of entropy that comes from releasing water. A special type of those like quote unquote hydrophobic interactions or nonpolar interactions that you'll see is going to be like pi stacking. So we have pi stacking. Where basically what happens is that in a pi bond, in one of these, when you have like an aromatic ring, the electrons are kind of concentrated above and below the ring. And so what can happen is that they can kind of sync up their electron clouds when they're stacked on top of each other. So if you think about there being another one in between, they can kind of like sync up their rings, and then you can get this like pi stacking interaction where these are going to kind of attract to one another based on those like dipole things. And so when we talk about the aromatic, this we can also think about pi stacking, which would be one of our like hydrophobic interactions, one of our nonpolar interactions, but it's going to be between like above and below the plane of these aromatic rings. So that's going to be a special type of one form of those interactions that we'll see. And we can also see that they can make these interactions, say, with the bases on nucleic acids. So we see that we have a lot of different options, but we can kind of, and we're going to have a lot of different protein structures. But thankfully, we can kind of simplify things because we can classify them in various ways, and we see the same sorts of repeating patterns throughout them. There's two kind of main types of proteins, I guess. I don't know how they call it, protein structures. So we have the fibrous, and then we have the globular. And so on this page are some examples from your book of the fibrous type. So we can talk about keratin. We can talk about silk and collagen. So we looked at collagen a bit when we talked about the secondary structure and how it had that weird like left-handed triple helix as opposed to our normal like right-handed alpha helix. This one had like 3 of these strands that are like have these weird like hydroxyproline. So there's one of those post-translational modifications coming in. So that would be an example of a post-translational modification. We'll talk more about these. Sometimes we call this a PTM. And remember, that's going to give it special properties. And in the case of this collagen triple helix, it's going to help make this structure. And it's why if you don't have enough vitamin C, you can't make those post-translational modifications, and you make your collagens all weak. And so then you get scurvy. And so, yeah, so your book goes in a little bit of that if you're interested. But basically, this is an example of a fibrous protein. You basically have this kind of like repeating units that kind of link together, and they make this, and they join together and they make some sort of structural protein. The other type that we're going to be talking about, which we're going to be mainly focused on, are going to be these globular. This is going to include things like enzymes and other proteins that have more like functional roles rather than just like structural roles. And there's going to be a lot of different ones, a lot of ones. But thankfully, we can kind of, just like we had those core secondary structures, we're going to see that we have core kind of repeating things that we see, these same sort of structural features that we're going to see in a lot of different proteins, often because they're related evolutionarily or at least functionally. So we have those, remember that we have those core secondary structure units. And we talk about tertiary structure, well, now we're just building upon that secondary structure, right? So we're going to have to incorporate those secondary structures into a larger structure, such as by, say, in the center of picture here, we have this these beta sheet in front of alpha helices. And so this would be an example of some sort of tertiary structure because we're involving side chain interactions. We're involving interactions between these different secondary structure elements and putting them together to make a larger fold. So we have those basic secondary structures that can kind of join together to form what we call super secondary motifs. So it's the word super secondaries because it's like above the secondary, but it's not like it's hierarchically like you go from secondary to super secondary to tertiary. No, it's part of the tertiary structure. So we call it super secondary in street. So even though we call it like super secondary, it's really a form of tertiary structure. So there's a bunch of different types of them. There are things like zinc fingers and beta turns and helix turn helix. From that, you could probably think of, okay, we have a helix, then we have a turn, and we have another helix. So we're putting together these secondary structural units to get these motifs. These motifs may or may not have a known function. For example, zinc fingers, they typically bind DNA. So here's an example of a zinc finger. You can see that it has an alpha helix, it has a beta strand, and it also has a coordinated zinc molecule. So that's one of the places where you can see that not only are proteins getting involved, but actually the other molecules that they're kind of bound to. And this is going to give it this characteristic shape that's going to allow it to bind to DNA. Now, motifs, we'll talk more about in a second about this word domain, but motifs can be located inside of a larger domain. So the way I like to kind of think about it is like protein motifs are a bit like furniture in a room. So it might be a chair or it might be a desk, or it might be a table. These may or may not have a known function, but they're often evolutionarily conserved and shared by many proteins. So basically, I might have a chair in my house, and you might have a chair in your house, and the guy next down the street might have a chair in his house. So basically, motifs can be found in a lot of different places because they're functionally useful, and because they're just kind of like smaller structural units that are made up of those core secondary structures. And because we have those core secondary structures that are shared between different proteins, well, you kind of expect that motifs would be as well, right? In terms of how these motifs are made up, well, we have to think about how can we connect those secondary structures. So say if we talk about those beta strands, so remember when we have individual ones, that's a strand, and then when they join together, they hydrogen bond to one another, we get these beta pleated sheets. If we have them anti-parallel, we can connect them with just short little turns, which is like those beta turns that we talked about. But if you have them parallel, well, now you have to have some sort of loop or region in between them. And so there are different ways that you can then connect them. What you'll see is that you'll often see this sort of like right-hand twist in these structures. And this is going to make it so that typically the connections are also going to be right-handed connections. And so this isn't something I'm going to hold you to remember, but like, see, if you look at this beta pleat sheet, you can see it kind of has this rightward twist. When we think about things like our barrels, we're going to see this right-handed twist. So here's an example of a beta barrel. Sometimes you'll see these making like channels through membranes, where you get this kind of these pleated sheets forming this circle. You also get things like a beta alpha beta loop. And so again, I don't need you to I want you to know a beta barrel, but I don't need you to memorize like a beta alpha beta loop. But you should be able to look at this and see, okay, well, we've got a beta strand, which is working with this other beta strand. So we've got a beta sheet, right? And then we've also got an alpha helix. So because of the way that this is actually laid out, we have a beta strand, we have an alpha helix, and then we have another beta strand. And so here you can see that although sometimes people talk about secondary structure as being between the adjacent regions on primary sequence wise, this could actually be pretty far away in primary sequence if you had something longer in between. But typically with a motif, we're talking about something shorter, something like this. And then these motifs can actually join together to give you a larger motif. So this is an example if we were to say, take that loop, well, now we can incorporate it into an alpha beta barrel. And so there are different ways that we can kind of join together these secondary structure elements to give us these super secondary elements, these motifs that can then join together to form larger motifs. Now, sometimes these motifs are going to be really, really small. So we looked at the zinc finger. That was an example here. We have a zinc finger. Sometimes we're going to have bigger motifs, and sometimes these motifs are going to have structures, functions, like known functions, and sometimes they're just kind of more of a shape. And when we're going to look a lot at this site, the PDB, which is going to have examples, it's going to be where they hold all the structures of these all these structural models that we're talking about, such as the one that I just showed you. And we're going to look and see how we can actually use the PDB to look at those structures. But basically in the PDB, and we'll take a look in a second, you can actually search for different structural motifs and see where they occur in all sorts of different proteins. I also recommend that you check out the PDB 101 site, and I'll put a link to it in the resources tab and things, but basically they have great explanations of how you actually go about using and interpreting the PDB. So those are our motifs, and now let's talk a little bit about domains. So if motifs were like our furniture, domains are like our rooms. So sometimes these can be distinct rooms, like if you're in an apartment that has actual doors between your rooms, like I finally am. So think of wall-separated rooms. But they can also be places in the house where you do specific things. So these can be functional domains, which may or may not be actually separated. So in my old apartment, when I had an efficiency apartment, and it was like one tiny little room where the bedroom was the same as the kitchen, which was the same as the living room, and it was all just one tiny little room. I would do different things in the different parts of the room. So I would sleep in my bed, and I would make food in the kitchen, quote unquote kitchen, which was just like a counter with a fridge and a little microwave across the way from my bed. Those would be kind of like different functional domains. They're all still kind of like within that same area, but I do different things in them. And then my bathroom, well, that actually was separated. And so that would be like a structural domain, but it would also have a specific function. So we talk about a protein domains, we can either talk about them as kind of distinct structural domains, distinct structural regions. Often we think about them as like being able to fold independently and function independently of the rest of the protein, but that's not always true. And sometimes we can talk about just like functional domains. Basically, domain is just a way that we can talk about a region of a protein, because we need some sort of way to refer to a region of a protein that we want to talk about. And often these domains are going to be things that are going to be shared throughout many proteins. But they're often going to be they're going to be containing multiple motifs often, and they can be these like larger regions. So let me just give you an example. So we'll start with a silly example, and then I'll go into an actual example. But if you think about a Swiss Army knife, well, different parts of it carry out different tasks. So you might have that cork part that's going to be able to uncork a bottle of wine. And you might have that bottle opener part that's able to open your beer bottle. And you might have that knifey part, which is able to slice open an envelope. So we could talk about those as different domains. So here they're kind of like structural domains. If I'm assuming that if we took that cork out, still be able to hold its shape. They're also functional domains because, well, they're doing different things. And so we could refer to, say, your cork domain of your Swiss Army knife. And so next time you need to open a wine bottle, I recommend you ask your friend for the cork domain of their Swiss Army knife. But anyway, this is true for proteins too. So they often have domains that are dedicated to certain tasks, but these tasks are often more difficult problems. One of these we're going to look at is carried out by a polynucleotide kinase phosphatase. So a lot of big words, but basically what it does is it's responsible for helping your repair broken DNA. So say there's some sort of break in DNA, which can happen such as from ionizing radiation or things like this. And often this creates these breakages where you have a 3' phosphate group and a 5' OH, which isn't going to be able to be fixed by DNA ligase. And so by DNA ligase, this is basically responsible for stitching together DNA, but it can only stitch together if you have a 3' OH and a 5' phosphate group. And so what's going to happen is that you need to convert what was the 3' phosphate to an OH group, and you need to convert that 5' OH to a phosphate group. Now, when we talk about dephosphorylating something, well, that we need a phosphatase for that. And if we talk about phosphorylating something, we need a kinase for that. And so we need a protein that's going to be able to do both of those things. And it needs to be able to recognize the site. And so what can happen is basically this one molecule is able to do it all by incorporating those different functions. And so it has these different domains. It has a kinase domain, which is able to do the phosphorylating. It has a phosphatase domain, which is able to do the dephosphorylating. And it has an FHA domain, which is able to find and bind to the site of the damage. And so I don't need you to remember all these details or anything like this. I just want you to see how different parts of a protein can do different things. And so this paper, they went and they actually looked and they solved the structure. So they figured out what it looks like, what all those domains look like. And when you talk about domains, you often see them displayed in different colors when you're looking at a structure. So we can see that the kinase domain is colored in yellow, the phosphatase domain is colored in blue, and the FHA domain is colored in green. And if you were to go and look at the sequence, they're often going to be colored that way as well. And here they're actually aligning the sequence of different polynucleotide kinases to show that these domains are actually evolutionarily conserved and the sequences are pretty similar. So this, you can see here too, that these pretty much look like structural domains as well as functional domains, right? So they look like they're kind of like separated in terms of their, like where they're located in the protein. And you could probably even separate them and study them alone, which they actually do. You also commonly see domains laid out in this sort of like topology map, where here it's emphasizing the secondary structural elements and how they're connected. So basically you can see you've got going from N terminus to C terminus, you have a beta strand, you have an alpha helix, you have a beta strand, you have alpha helices. So you can see kind of the layout of the protein and where the different secondary structural elements are laid out. Domains can contain motifs. So remember that your bedroom can contain a bed, your living room can contain a table, your bedroom could also contain a table. So domains can contain multiple motifs, and different domains can contain the same motifs. So one of the motifs that's present in this molecule is going to be this Walker A and B ATP binding motif. Again, I don't need you to know the names or anything like this, but basically this is a site that is found in a lot of different proteins that's able to bind to ATP because a lot of different proteins need to bind to ATP. And so you have this common motif that occurs that enables these proteins to bind to ATP. And it needs to bind to the ATP in order to carry out its kinase activity and actually transfer the phosphate group from that ATP onto the DNA. These domains are often conserved between related proteins. So here they're showing a comparison of the kinase domain of mouse polynucleotide kinase to T4 polynucleotide kinase, so this bacteriophage version that we often use in the lab to phosphorylate DNA. And so you can see that they're structurally conserved. What you can also see, hopefully, is that in this kinase domain, there's this like basic patch. So when we see blue, that's going to be basic. So think blue basic. And so that's going to be our plus charge region. And then if we have red, acidic, and that's going to be our minus charge. If we think about nucleic acids, well, they have that negatively charged phosphate backbone. So we would want a basic patch in order to bind them. And so that was one of the questions on your homework, is basically what would you expect to see in a DNA binding site? You would expect to have those basic amino acids, arginine, histidine, lysine, things that are going to make it positively charged so that it's attractive to that negatively charged nucleic acid. And so in that way, well, now you're able to create a binding site for this. And because it works so well, basically it's shared by a bunch of different molecules. It's been conserved over evolution. Speaking of evolution, I want to introduce this term homolog. Basically, a homolog is a gene or a protein that looks and or acts similar because they share an evolutionary origin. Don't confuse them with analog. So an analog is something that basically it looks similar or it acts similar, but it doesn't have a common evolutionary origin. So it can arise because of like convergent evolution. So different evolutionary paths, but they stumble upon similar useful things. So they look alike, but they're not actually like evolutionarily related, whereas homologs are. So here is an example. Here is the PDB entry for that example that we're talking about. And you can see here, you can see the Ramachandran outliers. Remember when I was talking about Ramachandran plots? You can see it here. So you want things to fall within the range of normal accepted angles, or there might be something that's weird with your experiments. But what I want to talk about now is if you go to the annotations. So annotations, that's basically when people put information on top of a sequence or on top of other things. They add information about things like domains, things like other known modifications, things like this. And so in here, there's actually a lot of different databases that hold like domains. And so there's different kind of classifications, but you can see that you can see information about the different domains it contains. So you can see there's the FHA domain, there's the phosphatase domain, the kinase domain, and things like this. You can also see like what types of folds it contains, as well as what it's related to. So like family, superfamily, all these various things. And so then you could go and you could check out where you see these different motifs found in other proteins. And so this is where you could do something like this. Another way that we see domains is often in this form of this domain architecture. And this is really great for highlighting evolutionary conservation. as well as highlighting the ways that different domains can be used in different proteins, in different species, in different proteins within the same species. And so you can see that the same domains can be used in lots of different types of proteins. And so sometimes, so the FHA domain, that's going to be the one that was going to recognize the damaged site. And you can see that, so basically that's going to recognize the phosphorylated site. And you can see that, well, sometimes it's hooked up so that when we looked at it, we saw it was hooked up to a phosphatase and a kinase. But sometimes it's just hooked up to a phosphatase, and sometimes it's just hooked up to a kinase, and sometimes it's hooked up to other sorts of domains. And so you can see that by kind of combining different domains, you can get different functions of proteins. And so evolutionary has had a lot to play with. And so we'll see these domains show up in all sorts of different contexts, in all sorts of different proteins. The key thing I want you to know is that these domains are going to be often they have specific structures, often they have specific functions. They can combine in different ways to give you different proteins with different activities. Now, in between, so when you see one of these domain architecture layouts, you'll see these domains that they kind of highlight, but then you also see these kind of other regions. Now, sometimes they just don't know what those regions do, but sometimes those regions might have like a structure and things, but often these are going to be IDRs. There's going to be those intrinsically disordered regions. And again, these regions might take a shape if they bind to something, and they might have really important roles in, say, like scaffolding and binding to other proteins and bringing them together. But they're not going to be like conserved typically, because there's a lot of different ways you can make a disordered thing, but fewer ways that you can actually make an ordered thing. One of the valuable things about having like domains and studying domains is that if we can figure out what the different parts do, we might actually be able to manipulate those parts in order to make proteins that do different things. Or maybe we can even, because domains are often structurally distinct, and can fold and act independently, we can actually study those domains in isolation and figure out exactly how they work and then modify them. Remember how I showed you that same domain that was like conserved with T4 polynucleotide kinase? Well, T4 is a phage, it's a bacteria infecting virus, and we use it in the lab a lot, and it's really, really helpful. And basically, you can buy mutants of it that have kinase activity, but no phosphatase activity. And this is great if you want to do things like radiolabeling RNA, so you can track it. And so I did that a lot in grad school. When I wanted to track RNA, I radiolabeled it. So I made it radioactive and then followed it. And so I added a radioactive phosphate group to it using this P4 polynucleotide kinase. Various reasons why these domains can be helpful, and studying domains can be helpful, and thinking about domains can be helpful. But remember that those domains are basically just regions of a protein, and they often, but not always, structurally independent. They may have a certain function, those rooms in your house. The motifs, those are going to be the furniture within your room, the smaller structural units that build upon the secondary structure motifs. Those build upon the secondary structure and combine those alpha helices and those beta strands in different ways to make these like common little structure motifs that you see in a bunch of different proteins. And all of those are coming down to those same interactions we started out talking about, those interactions between our side chains, between the side chains and the backbone, those interactions that involve things like our disulfide crosslinks, our ionic bonds. Remember, those are our salt bridges, our van der Waals interactions, our dipole-dipole interactions. All of these things, they're just happening between different regions of proteins to give us this structure. And so remember that those forces that could form were dependent on the amino acid sequence. And this is why the amino acid sequence, although we call it the primary structure, well, we call it that because it's of primary importance, not just because it's the first thing that kind of you see. But basically, that's going to help determine the secondary structure, which is going to help determine the tertiary structure. And so remember, with tertiary structure, we're talking with a single protein chain. But now let's go ahead and let's think about quaternary structure. And so remember, not all proteins will have quaternary structure, but some of them do. And so this involves when we have multiple protein chains that are acting together. And so if we basically have a single chain, it'll have no quaternary structure. So it acts as a monomer. So mono, one, monomer, one chain. But then we get oligomers and multimers. So when we see the word oligo, this is one of the ones on the core word roots terms I want you to know. Oligo, few. and multi, many. So if you have an oligomer that has a few subunits, a multimer, you have many. Typically, we often just like refer to, if we have an oligomer or whatever, we'll call it a dimer or a trimer or a tetramer. So 2, 3, or 4 subunits. And then we can also have pentamer, hexamer, all these different terms, depending on how many subunits we have. Then we can further classify multimers by whether the chains are the same or whether they're different. So if they're the same, then we call them homomeric. So again, going back to why it's important to know those roots. So homo is same, and we could have heteromeric, which would be different. So at least 2 of the chains are going to be different from one another. So we can talk about a homodimer or versus a heterodimer, or a homotrimer versus a heterotrimer. If they're all the same, we can call like the repeating unit. Each of these units would be, each chain would be a protomer. And when they join together, then that's when you get this multimer. And so those are different terms that you'll see used to describe these, describe the quaternary structure. Again, sometimes you might see disulfide bridges between the chains, but often it's just going to be non-covalent interactions. We'll see that this is going to come into play if we talk about how we might be able to separate these proteins with different experimental methods, and what would happen if you did or you didn't have a reducing agent, because the reducing agent would break up the disulfide bridges, but if you didn't have a reducing agent, then those chains would stay together. But don't worry about that yet, we'll get to that. We also get to hemoglobin, which it doesn't have those disulfide bridges in between them, but it's got some really, really cool features. And we're going to talk about hemoglobin, but basically it's a heterotetramer. So it has different subunits, but it has 2 copies of a beta subunit and 2 copies of an alpha subunit, at least for the adult version. And it's going to bind to iron and that will help bind to oxygen and carry oxygen throughout the bloodstream. Hopefully now you have a better understanding of what makes up a protein structure. Even when we talk about globular proteins, it's not just like a glob. It's not just like a weird looking thing. Well, I mean, some of them are kind of weird looking, but they're also really beautiful. And that beauty is related to its functionality as well. And so we have basically the amino acids are linking up. Remember that our sequence of amino acids is our primary structure. Now the backbone of those backbone of the amino acids interact with one another to give us our secondary structure, which involves that hydrogen bonding. So secondary, this is going to be our hydrogen bonding between the backbone. And then we have our tertiary structure. When we talk about what makes up our tertiary structure, well, then we can still have H bonding, but this time it would also be between the side chains or between the side chains in the backbone. We can also get ionic bonds. So that would be our salt bridges. We could get our disulfide crosslinks. And we can get our other van der Waals interactions. And so if we think about their strength or in their specificity, remember that the disulfide crosslinks, these are going to be strong and specific. On the other end of things, we have those like hydrophobic interactions, the nonpolar interactions. These are going to be generic, individually weak, but collectively they can be strong. Now remember, it's not an intrinsic desire for them to hang out with one another. They're just the losers that no one else wants to hang out with, so they get forced together. And then one of them tells a funny joke and they start kind of, okay, let's hang out. And so basically they're kind of stuck in place and they can get those instantaneous dipoles and things like this that can make them temporarily attracted to one another. And they're drawn together because of the hydrophobic effect, the exclusion of water, leading them to be clustered together, releasing that water to give you a gain in entropy. But those are going to be generic because all the amino acids can do that. With our ionic bonds, our salt bridges, these are going to be stronger than those, and they're going to be more specific. But they're not going to be as specific as your cysteine crosslinks, because remember, there's only a few cysteines in a protein. They have to be formed and survive and get out of the cell in the reducing environment of the cell, but they help certify things. The salt bridges, those are going to be more specific, but not as, they're more specific than our like van der Waals, but not as specific as the cysteine crosslinks. They're also going to be stronger than our van der Waals. They're going to be stronger than even our dipole-dipole interactions. And so we also have the hydrogen bonding was a form of dipole-dipole, so our polar. So H bonding, remember, that's just a special kind of dipole-dipole. And so when we have the polar amino acids, we can be thinking about the dipole-dipole, the hydrogen bonding, as well as the van der Waals. When we think about our nonpolar, we're just talking about those like other like transient van der Waals, induced polarity, things like this. And then when we talk about our ionic bonds, we're talking about our basic and acidic amino acids forming those salt bridges. Those are going to be more specific than the than just our other dipole-dipole or than our van der Waals, but they're not going to be as specific as the other ones because there's more of those acidic and basic amino acids, and they can also form other types of interactions and things like this. So those are not going to be as specific as our disulfide crosslinks. And then finally, we could have those interactions happening between the same chain, within the same chain, which would be tertiary structure, or within different chains, which would be quaternary structure. We can talk about within a tertiary structure or within a quaternary structure. We can talk about specific folds, specific motifs, versions, like little regions of a protein that contain various arrangements of secondary structure that are conserved between different proteins often. And you'll see them show up in a lot of places. And we'll also see how those different motifs are sometimes our furniture, those are sometimes contained within a bigger domain. A domain is kind of just a part of a protein that might be structurally and or functionally distinct. It's a region of the protein we just want to kind of like talk about. And often these domains are going to be shared evolutionarily between different proteins as well, but they might be hooked up in different ways to give you proteins with cool different functions. And so we'll look a lot more at this. But that's the overview of tertiary and quaternary structure.